So I was in the middle of explaining this diagram again. So the key is, yes, the mutated tRNA can recognize the stop codon, then the translation will continue. So at the end of 3' end of this tRNA, there will be an amino acid that's attached to it, so it's going to be delivered, maybe tyrosine, whatever that will be. Okay, so the translation will continue, so the nonsense mutation will be suppressed by the secondary mutation on the tRNA. That's what it's showing you. Okay. So next, let's talk about cancer for a little. So, so cancer is caused by the mutation mostly, because normal cells become cancer cells, and cancer cells have typically doing nothing but just replicating and growing instead of serving whatever the special function that those cells should serve. Okay, so that's the problem. And as the cancer cells are vigorously growing and taking over the space, then they will take over the space and the resources. And because of that growth of the cancer, people will have a problem and die from it. Okay, so then the question is, how do normal cells become cancer cells? So what's causing that? The change, okay, and normally the change is caused by mutations, okay, mutations on certain genes that will trigger normal cells become cancer cells. Okay, so then what are the mutations causing the cancer? There are a lot of studies going on. There are multiple genes that are known to cause the mutation. Okay, so then the question is then, would really the mutagenic condition, trigger the cancer development? The answer is yes. Okay, so-called the chemicals or conditions that causing cancer, we call them carcinogens. So carcinogenic conditions, right? So those are cancer-causing conditions, such as maybe toxic chemicals that will maybe induce mutations in normal cells. So normal cells will become cancer cells. Okay, and having said that, then yeah, most of the carcinogens are strong mutagens as well. So strong carcinogens, mutagens, kind of similar. Okay, so then you may wonder, so what kind of chemicals are considered as carcinogens and mutagens, right? And so we should know those, and we can avoid those because we don't have cancer developed in our system. Okay, so yes, actually there's a test for that, and especially based on Food and Drug Law, any companies manufacturing products, maybe the food, maybe cosmetics especially, or drugs, they're supposed to do a test called Ames test, which will use cell lines, especially bacterial cell lines. And you're going to expose those maybe bacterial cell lines to the product. If it's a cosmetic or if it's a drug, those will be exposed to the cells, and then you're going to measure the mutation rate of those cells that are exposed to your product and see if they will have a higher mutation rate. Okay, so actually what you're going to count is not actually the reversion rate, the secondary mutation rate. So I'm going to explain how that test, Ames test, works. Okay, so the key here is this test is used to screen maybe drugs or maybe cosmetics for their potential carcinogenic effect, in parentheses mutagenic effect. Okay, carcinogen, mutagen, I told you they are kind of similar. Okay, it's a kind of very quick and cheap test, because it doesn't require animals. Instead, you only use salmonella bacterial cells for this test. Okay, so how does that work? So we have mutant salmonella cells. Okay, especially those mutants are called His minus mutants. Okay, histidine, and His stands for histidine. Histidine is one of the amino acids that's very important. Okay, so normal salmonella, wild-type salmonella cells can make their histidine. Okay, they can synthesize them, but this mutant strain of salmonella, His minus, it has its unique mutation that will make this salmonella cell cannot synthesize histidine by its own. So the biosynthetic pathway of histidine is compromised by this mutation. Okay, so that's what his minus mutant means, that his minus symbol shows the phenotype which is disabling synthesizing histidine. Okay, that's what his minus mutant is. Okay, so that mutation is created through maybe base substitution or frame shift. Okay, so maybe one functional enzyme is disabled within these cells. So Salmonella type murine is the name of the bacterial cells, genus and species name. So anyway, this mutation, mutant cell line, these bacterial cells will be used. So these his minus mutant cells will be exposed to your maybe drug product or cosmetic product, and then you will measure the frequency of reversion mutation on those cells, his minus cells. Since it's a reversion, what kind of phenotype would you look for? So his minus phenotype will be changed to his plus phenotype, right? So yeah, so that's what you're going to measure. So this is the test setup. So you will have a test tube with the cells. Maybe S9 is that particular Salmonella cells. Okay, you'll have a number of those cells, about 10 to the 8th cell. That's about what, 100 million cells of his minus Salmonella cells are growing in there. And to that, you're going to add rat liver extract, rat liver. Why do you need rat liver? You know, so you're sacrificing an animal. So the rat is a mammal, just like human, right? And then they have a liver. So if this drug is maybe taken by a human, for example, normally drug ended up being in the liver, the detoxifying organ. When drug is taken by the human body, normally that's where they end up mostly. So you are kind of simulating inside of your body, okay, by adding some red liver tissue. Okay, there's a red liver tissue, and then Salmonella cells. And then, of course, your testing subject, okay, so whatever the drug or cosmetic testing, okay, will be added, okay. So they are mixed up, and then they'll be plated on a petri dish medium, okay. In this medium, the nutrients for the Salmonella cells to grow is added there, okay, except for histidine, okay. Histidine, I guess, is not included there, okay. So if it is the original cell, his minus cell, would they be able to grow there? Probably not be able to grow there because there's no histidine there. So the cells need histidine to be able to grow, right? So if there are any cells growing, okay, that means maybe those are the reverted cell, because you know, maybe through so many mutations happen. Some of them was a reversion mutation, so they recovered their phenotype of making their histidine in their cells, okay. So the reverted mutants can grow on this plate that doesn't have any histidine added there, okay. So you can count the number of cells that can show up on this plate, okay. So number of cells will give you the frequency of the secondary mutation, the reversion rate. Okay, so of course, any kind of experiment you perform, there should be a control. Okay, in this case, there will be a control made up with this same number of cells, this minus cell, and then rat liver extract will be added there. But that control will not be added with your testing drug. Instead, it will be added up with maybe just water, maybe deionized water. Okay, water shouldn't be any carcinogenic or mutagenic, right? So that's going to be used as a maybe negative control. Okay, so control is there. So that control treatment will be also plated on a plate or without histidine. Okay, so you'll compare the result, reversion result, between your control and your experimental setup, which has your testing drug in it. Okay, and see which one gives you higher rate of reversion. That's how this Ames test works. Okay, so normally the expected result is naturally, I mean, mutations happen inside of a cell. Okay, so naturally 5 to 10 reversions normally found even from the control. The cell is exposed to deionized water. Okay, so compared to that, okay, how many more reversions do you see? Okay, do you see more, maybe hundreds of reversions, or do you see the same amount? Okay, so that's what you're going to compare. If you see maybe tenfold increase on the reversion rate, of course that means yes, your drug that you tested strongly carcinogenic. It's increased, 10%, or maybe 100%, 10%, whatever the increased rate is. It's that much carcinogenic or mutagenic. Okay, so that's how you can measure it. So understand how Ames test works. Okay. Okay, now we're going to talk about the changes of the nucleotide, especially the base structures, how they are made under different mutagenic conditions. Okay, so DNA damages on base structures. Okay, we'll take a look at some examples that may be done spontaneously or through the induction, maybe through exposure to different chemicals. Okay, so if you expose your DNA to radiation, okay, strong energy, right, radiation energy, and or the mutagenic chemicals or spontaneous reaction, even with water, okay, so the base structure can be changed. So in the previous Ames test, even exposure to deionized water can induce the mutation. About 5 to 10 rebutants can be found, right? So that's the case. Okay? So spontaneous mutation is possible. So example is here, cytosine, okay, base can undergo spontaneous deamination. So amino group is removed. That's what deamination is. So NH2 is removed. Then the cytosine is changed to the structure. Okay. So if you compare that cytosine and then C without this amino group, and actually that structure is U. So deamination of cytosine will make that cytosine base as uracil base. Okay, so you understand the different base pairing capacity. C base pairs with G, but if it becomes U, actually DNA doesn't have any U. But if it's inserted through this mutation, then U pairs up with what? It's going to pair up with A, right? So that's a mutation. If U is used as a template, so it's going to bring in the A on the opposite strand. So that's a mutation. Okay, and then another example is adenine and guanine can be subjected to spontaneous deamination as well, which will convert adenine to hypoxanthine, different base, and then hypoxanthine can pair up with C instead of T. Okay, so A, if it's converted to hypoxanthine, it's going to pair up with C. Okay, and then if G is deaminated, and also it's going to become something called xanthine, and xanthine will pair up with C with 2 hydrogen bonds instead of the triple hydrogen bond. So it's going to make some changes. Okay, and then DNA also undergoes depurination by spontaneous hydrolysis of N-glycosyl linkage. Okay, what does that mean? So depurination means purine is removed. That's what it means. So the N-glycosyl linkage is the linkage that is holding the base to the phosphate sugar backbone, right? So yeah, purine, the entire purine can be removed. Okay, so it's showing up there. So the entire base can be removed. Then you will left with the sugar with the phosphate. That will be possible. If base is gone, then DNA polymerase has no idea what it can add. So it will create a mutation because the entire base is gone. Then polymerase doesn't know what to add there. Okay, and finally, so 5-methylcytosine can undergo deamination as well. So then if that happens, then it's going to generate thymine instead of cytosine. So C is switched to T. Of course, that's going to create a mutation. C base paired with G, but now T will base pair with A. So these are all possible damages that can happen on your base structure through a mutagen condition or mutation. Okay.